as you can see here. So take it away.
Yeah, so thanks for the introduction, Tom, and thanks to you and the rest of the organizing committee for inviting me to be part of this today. And it's great to be joining everybody online. And it's a sunny day here in Halifax too. So with Rizzy saying it's sunny out in BC, hopefully everybody from coast to coast is enjoying the same thing. We can all use a bit of extra sunshine in our lives right at the moment. So just to make sure we're all on the same page in terms of basic collagen structure, here's just a cartoon of the structural hierarchy of tendon. And of course, we know that way down over on the left, we have collagen molecules, which are long, slender, triple helical structures. And these come together in a very precise way to form semi-crystalline packing arrangements of collagen fibrils, which are long, thin, rope-like structures, generally between sort of 50 to, up to about 400 nanometers in diameter. Best seen using nanoscale imaging techniques such as electron microscopy or atomic force microscopy. And they've got the beautiful banding pattern on them because of that precise way collagen molecules are packed within them. Collagen molecules are linked within fibrils by collagen crosslinks, and when crosslinks initially form, they are divalent, which means that they link two molecules and they extend between lysine side chains from the telopeptide end of one molecule to the helix of a neighboring molecule. Now, with time, divalent crosslinks spontaneously react to form trivalent crosslinks, which link three collagen molecules together. And I'm just showing some of the example chemistries of these crosslinks. There's multiple different species, both of divalent and trivalent crosslinks. So these are just a couple of common examples here. Now, with further increases in time, we also have the formation of what are called non-enzymatic advanced glycation end product crosslinks, or AGEs for short. And these occur when sugar molecules react between lysine and arginine side chains, and they happen a little bit more haphazardly in a little bit less of a controlled way than the enzymatically controlled crosslinks form. Now, so this here is probably one of the best known figures relating collagen crosslinking to mechanics. What you see on the left is the change in cross-link profile with age, showing an initial decrease after birth in the ketoamine divalent crosslink HLKNL, and a corresponding increase in the divalent aldamine crosslink HLNL. And then later, the HLNL decreasing and being replaced by the trivalent crosslink HHL. And then, of course, later after that, the advanced glycation end product crosslinks starting to appear. And then on the right, what you see is change in mechanics with change in tissue age. And it's really easy to look at this figure and come away with the idea that the relationships between cross-linking and mechanics are very simple. Increased cross-linking gives rise to increased tissue stiffness and increased strength. And that's sort of, consistent with the biomaterials literature, which shows that exogenous cross-linking treatments, like glutaraldehyde in this case, gives rise to an increase in tissue strength and stiffness, markedly in this case. However, when we consider all of these plots that are on the screen now, there are some issues that we should be aware of. First of all, for those of you who are familiar with those first two plots on the left and have read the caption in the corresponding paper, you will know that they're actually from different tissues. So bovine skin collagen and rat tail tendon. And so there's no direct correspondence between them. And we also have some lack of clarity on how the cross-linking profile in rat tail tendon changes with age, and importantly, if that profile is representative of tissues that are relevant to human health. And then for the third plot, it's important to recognize that just because exogenous cross-linking treatments increase tissue strength and stiffness, that doesn't necessarily mean that the same is true for native cross-linking, as exogenous cross-linking can link structural levels that are not normally joined. So much of the work in our lab group has been investigating how collagen structure relates to mechanics. And a lot of our work, or a good bulk of our work, has used a bovine forelimb model as subject matter for exploring this. So the bovine forelimb contains these two functionally distinct tendons. On the palmar surface is the superficial digital flexor tendon, and it's a high-stress energy storing type of tendon that is used to transmit the forces needed to engage in locomotion. So it's got a maximum stress in life of about 70 megapascals. And on the dorsal surface of the forelimb is the common digital extensor tendon. And the extensor tendon is a low-stress positional tendon that's largely active when the forelimb is unloaded off the ground. So it has a maximum stress in life of about 10 megapascals. So we've used hydrothermal isometric tension analysis to look at the cross-linking in both of these two tendons. And most of you probably aren't familiar with HIT analysis. So just looking at this picture, the photograph here, what we do for HIT analysis is simply take small pieces of tissue and clamp them at fixed length and then heat them in a water bath. Now, if we look at schematically what goes on during this process, and here I'm just representing the piece of tissue as a series of triple helical collagen molecules linked together by crosslinks and held at the fixed length there by fixed supports with a load cell in series. When this is placed in hot water, at some point those triple helical collagen molecules will rupture the hydrogen bonds holding the alpha chains together, and they will denature. And when those three alpha chains separate in each of the molecules, the alpha chains are entropically driven to coil randomly, but are prevented from doing so by their isometric constraints, so they will generate measurable tension. With further heating, the individual alpha chains will start to undergo peptide bond hydrolysis, and the level of tension produced by that tissue will start to decay. And the rate of decay is related to the amount of cross-linking, with more cross-linked tissues undergoing slower load decay. So unsurprisingly, when we do this test for the high-stress flexor tendons and the low-stress extensor tendons, we find that the high-stress flexor tendons have significantly slower load decay, indicating significantly more cross-linking. That makes sense. The tendon that sustains high stress in life should have more cross-linking. What's surprising, though, is when you look at the mechanical response of these two tendons, it's the low-stress extensor tendon that is actually the stronger of the two. So that's really interesting. And we've also seen this in other tissues. So recently we looked at the differences between the posterior and anterior annular wall of ovine lumbar intervertebral discs, where the thinner posterior annulus sustains higher stresses than the anterior annulus. And like the bovine forelimb tendons, the posterior annulus has greater intermolecular connectivity as evidenced by slower load decay in HIT. And again, like the bovine forelimb tendons, contrary to what might be expected, it's those less cross-linked anterior samples of annulus that are actually the stronger. So to further investigate how differences in cross-linking affects mechanics, we've used an atomic force microscope to perform tensile stress strain experiments on single isolated collagen fibrils, testing them in a bowstring type morphology. And here on the screen in the picture, you can see a fibril pulled out in that bowstring conformation with the AFM tip that's being pressed into the surface, pulling the fibril out in tension. And we do this until the fibrils rupture. So here's a representative stress strain curve for a fibril from one of those high stress flexor tendons. And here's a stress strain curve for a single collagen fibril from the low stress extensor tendons. So during the initial loading of both fibril types behave similarly. No statistical differences here in the slope of those initial lines or the initial modulus or the stress at which the slope starts noticeably changing. And if Marcus is still in the session, he'll be familiar with this little picture from one of his papers here. So Marcus has done some great work showing what happens at the molecular level during initial loading. So we know during this initial phase of loading from 0% strain to 10% strain, extension is largely driven by the straightening out of the collagen triple helix. Now, from 10 to 20% strain, both fibril types again behave similarly to one another. And knowledge of what goes on within the fibrils in this range of strain is given by X-ray diffraction evidence showing that extension here is dominated by sliding between adjacent collagen molecules. It's only in the final portion of their loading curves that strains greater than 20% where the responses of the two fibril types actually differ. Here, the increased cross-linking in the flexor fibrils appears to impede molecular sliding, giving rise to a triphasic stress-strain response with a late increase in modulus. Now, when you take the fibrils after testing after they've fractured and re-image them with AFM, you see pretty dramatic differences in the rupture morphologies. So the limited molecular sliding of the flexor fibrils really seems to lead to a brittle type fracture. Whereas the more mobile molecules in the positional extensor tendon fibrils undergo significant discrete plasticity and the associated production of significant quantities of denatured collagen with that. So we've also investigated the impact of advanced glycation end product cross-linking on tendon mechanics using ribose incubation to induce AGE cross-linking. And this work was done on bovine tail tendon, which is also a low-stress positional tendon like the extensor tendon, and its fibrils also undergo significant discrete plasticity when ruptured. Now, ribose cross-linking, when done on these tendons, completely inhibits the discrete plasticity mode of of failure. So it really seems to restrict that molecular sliding ability from those added advanced glycation end product crosslinks. What's interesting, though, is when you look at the mechanics for both the treated and untreated tendon, there's no difference within the normal range of physiological loading. So through the linear portion of the stress strain curve below the yield point. Similar for both treated and untreated tissues. It's only in the overload region where that added advanced glycation end product crosslinking makes a difference to mechanics. So to wrap things up here, what does all this say about the figure that we started with? Well, clearly changes during tissue development impact the elastic region, and it's reasonable to assume that these changes are caused by increases in crosslinking. And after all, we know that if we inhibit crosslinking by limiting the enzymes required for it to occur, we see the opposite effect. However, what's important to realize is that the effects of altered enzymatic or advanced glycation end product crosslinking only appear to affect normal loading response in poorly cross-linked tissues. Meanwhile, for reasonably cross-linked tissues, cross-linking alterations mostly affect the overload region, which may have important functional implications for how native tissues and biomaterials respond to damage and cue repair. So with that, I am going to leave you with this beautiful image of our St. Mary's campus on the Halifax Peninsula, and thank all of the students who have contributed to the work in our labs, and my collaborators at Dalhousie, Drs. Laurent Kreplak, who played a role in the AFM work that I showed, and Michael Lee, who was involved with the ribose work, and also our funding sources. Thanks very much.
Thank you, Sam. Very nice work as usual.